Hello everyone. Welcome back to Microbiology Note. Today, we are diving deep into the fascinating world of microbiology to explore the bacterial growth curve. This curve provides a visual representation of how bacteria grow and adapt over time, and it's crucial for understanding their behavior in various environments. Whether you're a student, a researcher, or just curious about the microscopic world, this video will shed light on the different phases of bacterial growth and what they mean. So, let's get started and uncover the secrets of these tiny organisms. What is bacterial growth curve? The bacterial growth curve is a graphical representation of the different phases of bacterial growth over time, including the lag phase, log phase, stationary phase, and death phase. The bacterial growth curve is a graphical representation that illustrates the different phases of bacterial growth over time. It provides valuable insights into the growth patterns and dynamics of bacterial populations. The curve typically consists of four distinct phases: lag phase, log phase, exponential growth phase, stationary phase, and death phase. These phases reflect the changes in the number, size, and mass of bacterial cells during their growth cycle. Several factors influence bacterial growth, including temperature, pH, oxygen availability, nutrient availability, and moisture content. Bacteria are prokaryotic and unicellular organisms that primarily reproduce through a process called binary fission, where one parent cell divides into two identical daughter cells. To study the bacterial growth curve, certain measures need to be considered. First, a suitable culture medium must be provided to support the growth of microorganisms. As the bacteria utilize the nutrients in the culture medium, cell division occurs in a logarithmic or exponential pattern. Therefore, the growth of bacteria is typically expressed in logarithmic units rather than arithmetic units. This exponential growth results in a nonlinear graph, with the number of bacteria increasing rapidly over time. Distinct growth phases can be observed by incubating the bacteria at different time intervals. The lag phase is an initial period of adjustment during which the bacteria adapt to their new environment. In the log phase, bacterial growth is at its maximum, with the number of viable bacteria increasing exponentially. This phase is of particular interest in microbiological research and various applications. The stationary phase occurs when the rate of cell division equals the rate of cell death, resulting in a stable population size. Finally, the death phase is characterized by a decline in the number of viable bacteria, often due to nutrient depletion and accumulation of waste products. Measuring the growth rate of bacteria and studying the growth curve is crucial in microbiology. It is a fundamental technique used in basic research, as well as in agricultural and industrial applications. Understanding the growth patterns of bacteria helps researchers and industries optimize conditions for bacterial growth, such as in fermentation processes, food production, and the development of antimicrobial agents. By monitoring the growth curve, scientists can gain insights into the growth kinetics, survival strategies, and behavior of bacteria in various environments. Binary fission of bacteria. Understand the bacterial growth. Binary fission is the most common mechanism of cell replication in bacteria. It involves a series of steps that lead to the division of a single bacterial cell into two daughter cells. The process of binary fission begins with the cell undergoing growth and an increase in the number of cellular components. During this growth phase, the replication of DNA starts at a specific location on the circular chromosome known as the origin of replication. The circular chromosome is attached to the inner cell membrane. DNA replication proceeds in opposite directions along the chromosome until it reaches a region called the terminus. As the cell continues to grow, the center of the enlarged cell starts to constrict. This constriction eventually leads to the formation of two daughter cells. Each daughter cell receives a complete copy of the parental genome, ensuring that the genetic material is passed on accurately. Additionally, the cytoplasm is divided between the two daughter cells in a process known as cytokinesis. The protein FT plays a crucial role in directing cytokinesis and cell division. FT assembles into a structure called the Z ring on the cytoplasmic membrane. This Z ring is anchored by FT binding proteins and serves as a marker for the division plane between the two daughter cells. Other proteins essential for cell division are recruited to the Z ring, forming a complex known as the divisome. Once the divisome is fully assembled, it activates the production of a peptidoglycan cell wall and initiates the formation of a septum that divides the two daughter cells. The peptidoglycan cell wall is a key component of bacterial cell structure and provides strength and rigidity. Specific enzymes are involved in breaking the bonds between the monomers in the existing peptidoglycan layer, allowing the addition of new subunits along the division septum. During division, all of the outer layers of the cell, including the cell wall and outer membranes if present, must be remodeled to complete the process. This remodeling involves the coordinated action of various enzymes and proteins. Overall, binary fission is a highly orchestrated process in which a single bacterial cell replicates its genetic material, divides the cytoplasm, and forms two daughter cells. It relies on the precise assembly and activation of proteins such as FT and the divisome, as well as the remodeling of the cell wall and outer layers to ensure successful cell division. Requirements for bacterial growth. Chemical or nutrient requirement. Basics: bacteria need specific chemicals and nutrients for growth and metabolism. The lab culture medium supplies these essentials. Carbon makes up 50% of bacterial dry weight. Chemoheterotrophs derive it from organic materials like proteins and lipids. Chemoautotrophs and photoautotrophs get it mainly from CO2. Nitrogen, vital for amino acids, RNA, and DNA creation. Most bacteria use proteins, some, like nitrogen-fixing bacteria, get it directly from the air, while others use nitrate salts. Sulfur, needed for specific proteins and vitamins. Sources include proteins, hydrogen sulfide, and sulfates. Phosphorus, essential for DNA, RNA, ATP, and phospholipids. Derived from inorganic phosphate salts and buffers. Trace elements act as enzyme cofactors. Include iron, copper, zinc, and molybdenum. Oxygen, obligate aerobes, example, Pseudomonas, need oxygen. Obligate anaerobes, example, Clostridium, avoid it. Facultative anaerobes, example, Staphylococcus, E. coli, can adapt to both conditions. Aerotolerant anaerobes, example, Lactobacillus caries, don't need oxygen but can handle it. Microaerophiles need low oxygen levels but are harmed by high concentrations. Culture medium, used in labs to nourish bacteria. Its composition varies based on the bacterial species. Can be solid, semi-solid, or liquid based on agar levels. Physical requirements. pH levels. Bacteria have specific pH preferences. Acidophils thrive in acidic conditions, pH 0.1 to 5.4. Neutrophiles prefer near-neutral pH 5.4 to 8.5. Alkaliphiles favor alkaline conditions, pH 7 to 12 plus. Temperature. Mesophiles prefer moderate temperatures, 25 degrees Celsius to 40 degrees Celsius. Examples include E. coli and Staphylococcus aureus. Psychrophiles, cold-living, thrive between 0 degrees Celsius to 20 degrees Celsius. Found in polar areas. Thermophiles, heat lovers, flourish at 50 degrees Celsius to 60 degrees Celsius. Located in hot springs and geothermal zones. Osmotic pressure. Bacterial cells are about 80 to 90% water. Hypertonic solutions. High solute concentration causes water to exit cells, leading to plasmolysis and potential growth inhibition. Hypotonic solutions, low solute concentration can make water rush into cells, risking cell bursting. Some bacteria adapt to osmotic pressure extremes by producing osmoprotectants or regulating ion transport. Phases of bacterial growth curve. The growth curve has following phases: lag phase, log phase or exponential phase, stationary phase, death phase or decline phase. Now we will discuss about lag phase of bacterial growth curve. The lag phase of the bacterial growth curve is the initial phase after inoculation into a fresh medium where the population of bacteria remains temporarily unchanged. During this phase, there is no apparent cell division occurring, but the cells undergo various metabolic activities and preparations for growth. These activities include increasing in volume or mass, synthesizing enzymes, proteins, RNA, and other molecules, and adapting to the new growth conditions. The length of the lag phase depends on several factors, such as the size of the inoculum, time needed for recovery from physical damage or shock during transfer, synthesis of essential coenzymes or division factors, and synthesis of new enzymes necessary for metabolizing the substrates present in the medium. The lag phase is a period of adjustment for the bacteria to the new environment. While there is little to no cell division during this phase, the cells are not dormant. Instead, they are actively preparing for subsequent growth. The length of the lag phase can vary from 1 hour to several days, depending on the specific conditions and the bacterial species involved. At the end of the lag phase, the bacteria become fully prepared for cell division and enter the exponential growth phase of the growth curve. Log phase or exponential phase. The exponential phase is characterized by balanced growth, where all the cells are dividing regularly through binary fission and growing by geometric progression. The rate of cell division and growth is constant during this phase, depending on the composition of the growth medium and incubation conditions. The growth rate is expressed as the generation time, which is the time required for the population to double. During the log phase, the number of new bacteria appearing per unit of time is proportional to the present population. If growth conditions are optimal and not limited, the number of cells and the rate of population increase double with each consecutive time period. When the natural logarithm of cell number is plotted against time, it results in a straight line, and the slope of this line represents the specific growth rate of the organism. The specific growth rate is a measure of the number of divisions per cell per unit time. In the log phase, bacteria divide continuously at a constant rate, resulting in exponential growth. The bacteria are in their rapid stage of cell division and exhibit balanced growth. They have the smallest size during this phase. The metabolic activities, chemical composition of cells, and physiological characteristics of the bacteria are nearly uniform in the population during the log phase. This phase is often used to identify bacteria based on their biochemical and physiological characteristics. The generation time of bacteria is usually determined during the log phase, although it can vary among different bacterial species and growth conditions. It is the shortest during the log phase and is influenced by the growth factors present in the medium. The log phase can last for several hours, depending on the organism type, growth conditions, and population density. The exponential or log phase is a period of intense metabolic activity for microorganisms, during which primary metabolites are produced, enhancing the growth rate in terms of increased cell number and size. Microorganisms in this phase exhibit resistance to adverse conditions such as antibiotics and radiation. The number of cells produced exceeds the number of cells dying during the exponential phase. Overall, the exponential phase is crucial for the rapid growth and multiplication of bacteria, characterized by balanced growth, high metabolic activity, and increased biomass production. Stationary phase. The stationary phase is the third phase of the bacterial growth curve, following the log phase. The stationary phase occurs when bacterial growth slows down and eventually stops due to various factors such as nutrient depletion, accumulation of waste products, changes in pH, and limited oxygen availability. During the stationary phase, the rate of cell division is equal to the rate of cell death, resulting in a state of equilibrium. The number of new cells created is limited by growth factors, leading to a stable population size. Bacteria in the stationary phase undergo changes in their physiology and metabolism to adapt to the challenging environment. They enter a state of dormancy or quiescence to conserve energy and resources. Some bacteria in the stationary phase may produce secondary metabolites such as antibiotics. This is a survival strategy that can help inhibit the growth of competing organisms and enhance their own chances of survival. Bacterial cells in the stationary phase may also undergo sporulation, a process in which they form highly resistant spores that can survive harsh conditions and enable future growth when conditions become favorable again. The stationary phase is characterized by a decrease in metabolic activity and a slowdown in the synthesis of cell components. Bacterial cells become less susceptible to antibiotics and may show irregular staining patterns. Mutations can occur during the stationary phase, as DNA damage caused by endogenously generated reactive oxygen species becomes a major source of genetic changes. The stationary phase represents a critical stage in the bacterial growth cycle and is an important area of study for understanding bacterial survival strategies, antibiotic resistance, and other biological processes. If cells from the stationary phase are transferred to a fresh nutrient-rich medium, they can transition back to the log phase and resume their normal metabolic activities. Overall, the stationary phase is a period of adaptation and survival for bacteria when growth conditions become unfavorable, allowing them to persist until more favorable conditions arise. Death phase. The death phase is the fourth and final phase of the bacterial growth curve, following the stationary phase. During the death phase, the bacterial population declines as cells die off. This decline is primarily caused by two factors: the accumulation of toxic waste products and autolytic enzymes, and the exhaustion of available nutrients. In the death phase, the number of viable bacteria decreases continuously and exponentially. The total count of bacteria may remain relatively constant, but the viable count decreases as more cells die. The death phase is characterized by the presence of involute forms of bacteria. These forms may exhibit various shapes, such as long filamentous rods, branching or globular forms, or cells without a cell wall known as protoplasts, spheroplasts, or L-phase variants. However, when these involute forms are transferred to a fresh nutrient medium, they usually revert to their original healthy bacterial shape. The death rate during this phase is slower than the growth rate observed during the log exponential phase. Not all bacteria die at the same rate, as some may be more resistant and remain viable for a longer time. For example, spore-forming bacteria can withstand harsh conditions and persist longer than other bacterial cells. The death phase is brought about by various factors, including the depletion of essential nutrients and the accumulation of toxic waste products. As the nutrients become scarce and waste products accumulate, the conditions become increasingly unfavorable for bacterial survival, leading to cell death. If the incubation of the bacterial population continues after reaching the stationary phase, a death phase follows. During this phase, the viable cell population declines geometrically or exponentially, which is essentially the reverse of the exponential growth observed during the log phase. Autolysis, the process of self-destruction, occurs during the death phase. Bacterial cells break down and release their contents into the environment, contributing to the accumulation of waste materials. The death phase is influenced by factors such as temperature, nutrient availability, oxygen levels, and other injurious conditions. The rate and duration of the death phase can vary depending on these factors and the specific bacterial species. In some industries, continuous cultures are maintained to sustain the logarithmic growth phase of cells by periodically supplying nutrients and maintaining the appropriate aerobic conditions. This allows the bacteria to avoid entering the death phase and prolong their growth. Growth rate and generation time. The growth rate and generation time of bacteria are important parameters that describe their reproductive capabilities. The growth rate refers to the speed at which a bacterial population increases in number during the exponential growth phase. It is commonly measured by determining the doubling time or generation time of bacteria. The generation time is the time it takes for the population to double in size. Under standard nutritional conditions, including the culture medium, temperature, pH, and other factors, bacterial growth rates vary, leading to different generation times. The generation time can range from as short as 12 minutes to as long as 24 hours or more, depending on the species and environmental conditions. For example, in the laboratory, Escherichia coli, a commonly studied bacterium, has a generation time of 15 to 20 minutes. However, in the intestinal tract, the generation time of coliform bacteria like E. coli is estimated to be 12 to 24 hours. Most cultured bacteria have generation times ranging from approximately 15 minutes to 1 hour. Certain bacteria, such as symbionts like Rhizobium and lithotrophs like nitrifying bacteria, tend to have longer generation times compared to others. Pathogenic bacteria like Mycobacterium tuberculosis and Treponema pallidum have particularly long generation times, which is believed to contribute to their virulence. The generation times of various bacteria under optimal growth conditions are listed in below. Escherichia coli, 17 minutes in glucose salts medium. Bacillus megaterium, 25 minutes in sucrose salts medium. Streptococcus lactis, 26 minutes in milk. These values provide insights into the relative growth rates and generation times of different bacterial species under specific growth conditions. Understanding the growth rate and generation time of bacteria is important for various applications, including microbial ecology, industrial processes, and the development of treatment strategies against bacterial pathogens. Significance of the bacterial growth curve. The bacterial growth curve holds significant importance in various fields, both in research and commercial applications. Understanding the growth kinetics and population dynamics of bacteria provides valuable insights and aids in decision-making processes. Here are some key aspects highlighting the significance of the bacterial growth curve. Utilization and inoculation. Bacterial growth curves are crucial when it comes to utilizing or inoculating known numbers of bacterial isolates for specific purposes. For instance, in agriculture, understanding the growth curve helps in enhancing plant growth by introducing beneficial bacteria. Similarly, in biodegradation processes, such as the breakdown of toxic organics, knowledge of growth kinetics is essential for optimizing the inoculation of appropriate bacterial strains. Industrial production. Bacterial growth kinetics play a vital role in industrial production. By studying the growth curve, scientists and industry professionals can determine the optimal conditions and nutrient requirements for producing antibiotics or other natural products on a large scale. This knowledge helps in maximizing production efficiency and yield. Substrate metabolism. Bacterial growth curves are useful for assessing the metabolic capabilities of bacterial strains. By studying the growth kinetics, researchers can determine if specific strains are adapted to metabolize certain substrates. This is particularly valuable in areas such as waste management, where bacteria are engineered to clean up pollutants. Growth curves can indicate whether the bacteria can effectively metabolize industrial waste or oil pollution without being hindered by toxic effects. Evaluation of energy sources. The slope and shape of growth curves obtained from bacteria grown with mixtures of industrial waste products provide insights into their ability to metabolize particular substances. This information helps scientists assess the potential energy sources available in the waste mixture and determine the suitability of specific bacterial strains for waste treatment or bioremediation purposes. In summary, the bacterial growth curve is a powerful tool that enables researchers and industry professionals to understand the growth kinetics, population dynamics, and metabolic capabilities of bacteria. This knowledge is crucial for optimizing processes, developing sustainable solutions, and utilizing bacteria in various applications, ranging from agriculture to waste management and industrial production. Factors affecting bacterial growth curve. Nutrient availability. Bacteria require essential nutrients such as carbon, nitrogen, vitamins, and minerals for growth and reproduction. The availability and concentration of these nutrients in the environment can significantly impact the bacterial growth curve. Limited or inadequate nutrient supply can slow down or halt bacterial growth. Temperature. Bacteria have an optimal temperature range for growth, and deviations from this range can affect their growth rate. Higher temperatures can denature proteins and inhibit enzyme activity, leading to decreased growth. Low temperatures can slow down metabolic processes and delay bacterial growth. Each bacterial species has a specific temperature range at which it thrives best. pH, the acidity or alkalinity of the environment, expressed as pH, influences bacterial growth. Bacteria exhibit different pH requirements depending on the species. Changes in pH can disrupt enzyme activity and cellular processes, affecting growth. Some bacteria prefer neutral pH, while others thrive in acidic or alkaline conditions. Oxygen. The availability of oxygen in the environment has a significant impact on bacterial growth. Some bacteria require oxygen, obligate aerobes, for their metabolic processes and growth. Others are unable to tolerate oxygen, obligate anaerobes, and may even be harmed by its presence. Additionally, certain bacteria can grow under both aerobic and anaerobic conditions, facultative anaerobes. Moisture. Adequate moisture is essential for bacterial growth. Bacteria typically require a certain level of water activity (AU) in their environment to carry out metabolic processes. Insufficient moisture can lead to desiccation and inhibit growth, while excessive moisture can create unfavorable conditions or favor the growth of other organisms. Environmental factors. Various environmental factors, such as light, pressure, and radiation, can impact bacterial growth. Some bacteria are sensitive to light and can be inhibited by its presence, while others may require specific light conditions for growth. Pressure and radiation can also affect bacterial growth, either inhibiting or promoting it depending on the species and conditions. Inhibitory substances. The presence of inhibitory substances, such as antimicrobial agents or toxic compounds, can hinder bacterial growth. Antibiotics, disinfectants, or other chemicals may be present in the environment and can inhibit bacterial growth or even cause cell death. Competition. Bacterial growth can be affected by competition with other microorganisms for resources and space. In mixed microbial communities, certain bacteria may outcompete others for nutrients, limiting their growth. Micronutrients, including trace elements like iron, copper, zinc, and molybdenum, are essential for bacterial growth as they serve as cofactors for enzymatic reactions. Adequate availability of these micronutrients is crucial for B. Applications of bacterial growth curve. Antibiotic efficacy testing. The bacterial growth curve is utilized to evaluate the effectiveness of antibiotics and antimicrobial agents. By measuring bacterial growth over time in the presence of different concentrations of an antibiotic, researchers can determine the minimum inhibitory concentration, (MIC), required to inhibit bacterial growth. This information helps guide antibiotic treatment strategies and the development of new antimicrobial drugs. Biotechnology and industrial processes. Understanding the bacterial growth curve is crucial in various biotechnological and industrial applications. It helps optimize fermentation processes, such as the production of antibiotics, enzymes, biofuels, and other valuable products. By monitoring and controlling the growth phases, nutrient supply, and environmental conditions, researchers can maximize product yield and quality. Food safety and preservation. Bacterial growth curves are employed in food safety and preservation practices. By studying the growth characteristics of pathogenic bacteria in different food matrices, scientists can identify critical control points to prevent bacterial contamination and ensure food safety. Additionally, the growth curve aids in determining the shelf life and storage conditions for various food products. Microbial ecology. The study of microbial communities and their dynamics in different environments relies on the bacterial growth curve. By examining the growth patterns of specific microbial populations, researchers can infer their roles, interactions, and responses to environmental changes. This information contributes to understanding ecosystem processes, bioremediation, and the impact of microbial activities on the environment. Disease pathogenesis and drug development. The bacterial growth curve provides insights into the pathogenesis of infectious diseases and aids in the development of new therapeutic strategies. By monitoring bacterial growth in host organisms or in vitro models, researchers can identify critical growth stages, evaluate virulence factors, and assess the efficacy of potential drugs or vaccines. Pharmacokinetics and drug dosage. Pharmacokinetic studies involve understanding how drugs are absorbed, distributed, metabolized, and eliminated by the body. Bacterial growth curves assist in determining optimal drug dosage regimens by assessing the drug's effectiveness in inhibiting bacterial growth over time. This information helps guide dosing schedules and treatment protocols. Environmental monitoring and water quality. Bacterial growth curves are used in monitoring and assessing water quality and environmental contamination. By analyzing bacterial growth patterns, researchers can evaluate the presence of indicator organisms or pathogens, detect pollution sources, and assess the efficacy of disinfection or treatment methods. Alright folks, that wraps up our deep dive into the bacterial growth curve. I hope you found this video insightful and informative. If you're keen on diving even deeper and want study notes related to this video, I have got a recommendation for you. Head over to Google and search for microbiologinote.com. It's a fantastic resource packed with detailed notes that will complement what we have discussed today. Don't forget to like, share, and subscribe for more microbiology insights. Thanks for watching, and until next time, keep exploring the microscopic world.